Welcome back to Bookfulness, the deep dive where we explore the foundational concepts of evolutionary science. This is Deep Dive 9, and we are concluding Unit 2, which has been focused entirely on the fundamental mechanisms of evolution. You know, how selection, drift, mutation, and gene flow shape populations in space and time.
That's right. We've spent a lot of time recently talking about divergence, how populations split, adapt locally, how geography can change everything genetically.
Giving it a building block.
Exactly. But today, we're zooming out a bit. We're going to discuss the ultimate, well, often permanent result of that divergence.
Yeah.
The actual formation of new, distinct species.
Right. That moment when two groups become entirely separate evolutionary paths, independent lineages.
Precisely.
And our mission today is to explore Chapter 9, Species and Speciation, to understand not just, what a species is fundamentally, but how the barriers that actually define them evolve.
Yeah, it sounds simple on the surface, doesn't it, with a species? But as soon as you start digging, it gets complicated fast.
Deceptively simple, exactly. So let's start with that essential question. When does a population or a group of populations officially become a species? How do we draw that line?
Well, for a long time, centuries actually, the definition was basically just visual. Early naturalists, Linnaeus being the most famous.
The father of taxonomy.
Right. They categorized species essentially as groups of organisms that you could visually distinguish from one another. If they looked different enough, they were considered different species. Simple as that.
A neat, tidy system for cataloging life. Put things in boxes based on appearance.
Seemed logical. And it worked okay for a while.
Right up until biologists started, looking closer, observing populations in the wild, analyzing their genetics.
Exactly. And that simple visual yardstick, as the chapter points out right away, just doesn't hold up. It's, frankly, completely unreliable in many cases.
Proven unreliable time and time again, I imagine.
Oh, absolutely. And the perfect example to start with, the one the book uses to illustrate this, is the case of the North American screech owls.
Okay, let's unpack that one because it really highlights the paradox perfectly.
Right, so consider the eastern screech owl, Megascops azio. Within this single species, you find two really different color forms. There's a predominantly gray morph, and then there's this rich reddish-brown one.
Visually quite distinct. If you were just following Linnaeus's rule,
you might easily classify them as two different species, just based on that stark color difference.
But they don't care about the color difference, do they?
Not at all. They interbreed constantly, freely. A gray owl mates with a reddish-brown one, they produce offspring, no problem. It's clear they're just one species showing a pretty common genetic variation, a polymorphism likely controlled by just one gene locus.
Okay, so far so good. Appearance doesn't match breeding reality. Case 1.
Now, here comes the twist that really messes with the visual definition. We look west, and we find the western screech owl, M. kennicoti. And the critical thing is, this western owl looks almost identical to the gray morph of the eastern species.
Visually indistinguishable, even for experts.
Pretty much, yeah. Without looking really closely at subtle secondary traits, or, more importantly, hearing them, you'd struggle to tell them apart just by sight.
Okay, so based on looks, they should be the same species, or at least very closely related and potentially interbreeding.
You'd think so. But biologically, they are profoundly separate.
So what's keeping them apart if it's not their feathers? What are the barriers?
Well, the differences lie mainly in their behavior and their genetics. The western owl has a completely different voice, a different song pattern. It's more of a tremolo, whereas the eastern owl has this sort of descending whinny.
Ah, the sounds are different, like a mating signal difference.
Exactly. And where their geographic ranges overlap, down in Texas, for example, they show very little, if any, gene flow. They just don't seem to recognize each other as potential mates, likely because of that voice difference.
So the behavior acts as a wall.
A very effective wall. And furthermore, if you look at their genetics, especially the mitochondrial DNA, which is inherited maternally, it confirms they are entirely separate lineages. They've been evolving independently for a long time.
Wow. Okay, so let's recap. We have two groups, the eastern color morphs, that look completely different but mix freely. One species. And then we have two groups, the eastern gray and the western, that look almost identical but are biologically, reproductively totally separate. Two species.
That's the crux of it.
The conclusion seems unavoidable then. Physical appearance can be incredibly misleading. It's just noise in a way. The real distinction has to be biological. It has to center on reproduction, on who can successfully mix genes with whom.
And that realization leads us straight into the most influential concept in modern evolutionary biology for defining species, which was really formalized by Ernst Mayr back in the 1940s. It's called the Biological Species Concept, or the BSC.
Okay, let's dive into this definition properly because it feels like a fundamental shift. It moves away from just cataloging based on looks towards understanding the evolutionary process itself.
That's exactly its strength. So the BSC defines species as, I'm quoting loosely here, groups of actually or potentially interbreeding natural populations, which are reproductively isolated from other such groups.
Actually or potentially interbreeding, and reproductively isolated, those are the key phrases.
Absolutely. The focus is laser sharp. The defining criterion isn't what you look like or where you live or exactly what you eat. It's reproductive isolation. Can your group successfully exchange genes with that other group? If the answer is no, or significantly limited, then you are an independent species.
That seems powerful because it directly ties the definition to the evolutionary process. If you're reproductively isolated, you're on your own evolutionary trajectory. Your gene pool is independent.
That is the core insight. Reproductive isolation stops or strongly restricts gene flow between groups. And without that homogenizing effect of gene flow.
Yeah, mixing everything up.
Right. Without that, selection and genetic drift can operate independently on each group, driving them further apart genetically and ecologically over time. They accumulate their own unique set of mutations, their own adaptations.
So they become distinct evolutionary units. Makes sense.
But it's important to clarify something here. The BSC doesn't necessarily demand absolute 100% isolation. The source material is careful to point this out.
Oh, so a little bit of hybridization is okay.
Yeah, little genetic leakage through occasional hybridization might happen, and that's often acceptable under the BSC. The key point is whether that leakage is significant enough to prevent the two groups from evolving independently.
So if hybridization is rare, or if the hybrids themselves aren't very fit.
Exactly. If maybe only 1% of matings result in hybrids, and maybe those hybrids don't survive well or can't reproduce effectively themselves, then the two gene pools are still largely distinct. They're still following their own evolutionary paths. That reproductive barrier is strong enough, even if it's not perfect.
Okay, so it's about the effectiveness of the isolation in maintaining evolutionary independence. Hashtag, tag, 1.3 limitations of the BSC. Right, the BSC sounds conceptually very strong, very focused on the evolutionary process. But, I find the limitations almost as revealing as the concept itself. They really highlight the complexities, the messiness of biology in the real world. And the first big one is what the book calls the allopatry problem.
Ah, yes, the allopatry problem. This is probably the biggest practical headache for the BSC.
Explain that. What does allopatry mean again?
Allopatric just means geographically separated. So imagine you have two populations of, say, mice. One lives on one side of a huge canyon, the other lives on the opposite side. There's no way for them to cross. They're completely isolated by geography.
Okay.
Now, under the BSC, we need to figure out if they're potentially interbreeding. But how can we possibly know? They never meet in the wild because of the canyon.
Can you just bring them into the lab and see if they mate?
You can try that. But a lab is an artificial setting. Maybe they'll mate in a cage but would never recognize each other or find each other in nature. Or maybe they won't mate in the lab due to stress, but they actually could in the wild if they met. It doesn't reliably tell us about potential interbreeding in their natural context.
So because you can't observe potential interbreeding for geographically separated populations, you can't strictly apply the BSC to confirm if they are distinct species or just isolated populations of the same species.
Exactly. Unless you can somehow magically remove the canyon and watch what happens over generations, the BSC becomes more of a theoretical ideal than a practical tool for classifying allopatric populations. We often have to infer species status based on other things, like genetic distance or morphology, even if we conceptually prefer the BSC.
Okay, that's a big limitation for things living apart. What's the other major issue?
The other massive hole in the BSC's applicability is the asexual problem.
Organisms that don't have sex.
Precisely. The entire biological species concept hinges on sexual reproduction, on interbreeding, and the resulting gene flow, or lack thereof. But vast numbers of organisms on Earth reproduce asexually. Think bacteria, archaea, many protists, some plants, even some animals like certain rotifers or lizards.
They just clone themselves, essentially.
Yeah, right. They don't interbreed with anyone, let alone different groups. So the whole concept of reproductive isolation is meaningless for them. You simply cannot apply the BSC to define species boundaries in these huge, diverse groups of life.
So it works well conceptually for sexually reproducing organisms that live together, but struggles with geographically isolated ones and fails completely for asexual ones.
Okay, so if the BSC, which seems like the evolutionary gold standard, has these significant practical gaps, what do biologists, especially taxonomists who actually need to classify organisms, use instead? What's the alternative?
Well, in practice, many biologists, particularly those focused on classification and systematics, often rely on the phylogenetic species concept, or PSC.
Phylogenetic, relating to the evolutionary tree, the phylogeny? Yes, exactly. The PSC takes a different approach. It focuses purely on evolutionary history, as reconstructed in phylogenetic trees. It defines a species basically as the smallest distinguishable group of populations that forms a distinct branch on the tree.
So it's about diagnosability. If you can reliably identify a group as unique based on shared derived characteristics that may be unique DNA sequences or specific skeletal features or something, then it counts as a species under the PSC.
That's the essence of it. It looks for monophyletic groups, groups that include an ancestor and all its descendants that are identifiable by unique traits, often called synapomorphies.
That sounds incredibly practical. You can apply that to anything, right?
Absolutely. That's its major advantage. The PSC is universally applicable. You can use it for asexual bacteria, for those allopatric mice on opposite sides of the canyon, even for fossils where you only have morphology to go on. If you can build a tree and identify distinct clusters, you can define species. It's what largely drives modern naming and classification efforts.
Okay, so if the PSC is so practical and universally applicable, why do we even bother with the BSC? Why do evolutionary biologists, as you mentioned, still seem to favor the BSC conceptually?
That's a crucial distinction. It's about what you're trying to understand. The PSC is excellent for identifying and cataloging diversity, recognizing the products of evolution.
The branches on the tree.
Right. But the BSC, despite its limitations, aims to capture the process of diversification. Evolutionary biology is fundamentally about understanding how new species arise. And the central event in that process for sexually reproducing organisms is the evolution of reproductive isolation. That's the point when lineages truly become independent.
So the BSC focuses on the mechanism, the biological barrier that stops gene flow, while the PSC just identifies a pattern of difference.
Pretty much. The PSC might identify a distinct phylogenetic group that arose purely due to, say, genetic drift and isolation, without any strong biological barrier preventing interbreeding if they were to meet. Or you could potentially use the PSC to split a single large interbreeding species into many species based on very minor localized genetic variations that form shallow branches on a tree.
So the BSC, by focusing on reproductive isolation, tries to pinpoint the specific biological changes, the walls being built, that allow independent evolution to happen. That seems more fundamentally about the origin of species.
Exactly. The BSC forces us to ask the critical question: what biological feature prevents these two groups from merging back into one? Answering that question gets us closer to understanding the drivers and mechanisms of biodiversity than simply labeling branches on a tree. And that question, how do these barriers evolve, is what we need to tackle next.
All right, so if reproductive isolation is the absolute key to speciation under the biological species concept, then the whole process of speciation boils down to the evolution of these biological differences that prevent or reduce gene flow. We call these differences
Reproductive Isolating Barriers, or RIBes for short.
RIBes, okay.
And it's important to realize up front that achieving total reproductive isolation usually isn't the result of just one single dramatic barrier popping up.
It's not like one mutation instantly makes two groups incompatible, usually.
Yeah, not usually, no. More often, it's the cumulative effect of several different RIBes acting together, often in sequence. Think of it like layers of defense. If one barrier fails, another might kick in. Together, they ensure the gene pools stay largely separate.
Layers of resistance. Can you give an example of how these layers work in a real-world case?
Absolutely. The book uses a classic example. The two species of monkey flowers found in the Sierra Nevada mountains, Mimulus lewisi and Mimulus cardinalis.
Okay, monkey flowers. What's their story?
Well, these two species often grow near each other, sometimes side by side at intermediate elevations. One, M. lewisi, has pink flowers, and the other, M. cardinalis, has bright red flowers.
Pink and red. Got it.
Now, here's the interesting part. If you bring them into a greenhouse or a lab, you can cross-pollinate them quite easily.
They hybridize.
Yes, and they produce viable, fertile hybrids. Genetically, they're still compatible enough to produce successful offspring in that artificial setting.
Okay, so intrinsic genetic incompatibility isn't the main barrier here. Why are they still distinct species in the wild then? Why don't they just merge into one hybrid swarm?
Because of a sequence of powerful prezygotic barriers that act before fertilization even happens.
Ah, the first line of defense. What are they?
First, there's a degree of ecological separation. They tend to prefer slightly different microhabitats or elevations, but that's not absolute. The really crucial barriers relate to their pollinators.
Pollinators, how does that work?
M. lewisi, the pink one, has flowers with a wide opening, nectar guides visible to bees, and it's primarily pollinated by bees like bumblebees.
Okay, bee pollinated.
M. cardinalis, the red one, has narrow, tubular flowers with lots of nectar suited for hummingbirds, and it lacks nectar guides visible to bees. It's almost exclusively pollinated by hummingbirds.
Red flowers, hummingbirds. Got it.
So, even if the plants are growing right next to each other, the bee carrying lewisi pollen is very unlikely to visit the red cardinalis flower, and the hummingbird visiting cardinalis rarely probes the pink lewisi flower.
The pollinators act like specific delivery services, keeping the pollen separate.
Exactly. The differences in flower shape and color create pollinator isolation. This behavioral choice by the pollinators acts as a massive reproductive barrier. These prezygotic barriers, the slight habitat difference plus the very strong pollinator isolation, account for almost all the reproductive isolation we see between them in nature.
Wow. So even though they could make fertile hybrids, they rarely get the chance because the pollen doesn't get transferred effectively. That really shows the power of these initial barriers. Hashtag tag 2.2 prezygotic barriers, preventing fertilization. Okay, so that monkey flower example highlights prezygotic barriers. These seem really important because they prevent wasted effort, right? You don't invest energy in making a hybrid that might be unfed anyway.
Precisely. From an evolutionary perspective, preventing fertilization in the first place is often the most efficient way to maintain species boundaries. The book lists several major types, drawing from Table 9.1.
Let's run through the main categories. What's the simplest?
Maybe the simplest is temporal isolation. This just means that species breed or mate at different times.
Could be different seasons of the year, like one plant flowers in early spring, another in late summer.
Yep, or at different times of day. One species of coral might release its gametes at dusk, another releases them an hour after midnight. If their reproductive timing doesn't overlap, they simply never have the opportunity to interbreed.
Simple but effective. Okay, what else?
Then there's ecological isolation. This occurs when species live in the same broad geographic area, but occupy different habitats or niches within that area, so they rarely encounter each other during their mating periods.
So they're sympatric geographically, but ecologically segregated, like the monkey flowers having slightly different elevation preferences.
Exactly. Or the example given in the book about two Japanese species of ladybird beetles. They live in the same forests, but one species feeds and mates exclusively on thistles, Cirsium, while the other feeds and mates only on mayapple, Calophyllum.
Ah, so their host plant preference dictates where they mate.
Right. Since they only meet potential mates on their specific host plant, and they use different host plants, they effectively remain reproductively isolated, even though they might fly past each other regularly. The ecology creates a barrier.
Okay, timing and location. What about behavior? That seemed key for the screech owls.
Absolutely crucial, especially in animals. This is sexual or behavioral isolation. It involves differences in species-specific courtship rituals, mating calls, visual signals, or chemical pheromones.
Like the different songs of the screech owls.
Perfect example.
Yeah.
Or think about the elaborate dances of birds of paradise, the specific light flash patterns of fireflies, the chemical signals released by female moths to attract males of only their species. Often, females simply aren't stimulated by or reject the courtship signals of males from other species.
The communication just fails. They're speaking different mating languages.
You could put it that way, yeah. It's a very widespread and powerful isolating mechanism.
Okay. What if they get past the signals and actually attempts to mate?
Then you might encounter mechanical isolation. This is basically physical incompatibility of the reproductive organs.
The parts just don't fit.
Essentially, yes. It's often referred to as the lock and key hypothesis, especially common in insects where the male genitalia, the key, might only fit correctly into the female reproductive tract, the lock of its own species.
Like a physical barrier preventing sperm transfer.
Correct. Even if courtship occurs, if the physical structures are incompatible, fertilization can't happen.
All right, so time, place, behavior, physical fit. Is there anything else before fertilization?
Yes, one final prezygotic hurdle. Gametic isolation. This happens at the level of the sperm and egg themselves.
So mating might occur, sperm might be transferred, but fertilization still fails.
Exactly. The gametes are incompatible. This can happen in several ways. Maybe the sperm isn't viable in the reproductive tract of the other species, or, more commonly, the proteins on the surface of the sperm and egg that are required for recognition and binding don't match.
Like a molecular lock and key failure.
Precisely. The sperm might not be able to penetrate the egg, or the fusion process itself might fail. This is particularly important for species with external fertilization, like corals or sea urchins releasing gametes into the water. But it definitely occurs in internally fertilizing species too. Hashtag tag 2.3 postzygotic barriers acting after fertilization.
Okay, that's a comprehensive list of ways to stop fertilization from happening. Prezygotic barriers seem really dominant. But what if? What if all those fail? What if, despite different timings, habitats, signals, and gamete recognition issues, a sperm from one species does manage to fertilize an egg from another species? A hybrid zygote is formed.
Right. Then we move into the realm of postzygotic barriers. These barriers act after fertilization has occurred, and they function by reducing the fitness, the survival or reproductive success of the hybrid offspring.
So the hybrid is formed, but it's somehow deficient.
Yes, and we can broadly categorize these postzygotic barriers into two types based on why the hybrid has low fitness. Extrinsic and intrinsic?
Extrinsic and intrinsic. Okay, what's extrinsic isolation?
Extrinsic isolation means the hybrid itself might be perfectly healthy biologically, but it has reduced fitness because it's poorly adapted to the environments of either parent species. Its traits are intermediate, and that intermediate state just doesn't work well out there.
Can you give an example?
Sure. Imagine two fish species, one adapted to fast-flowing water, one to still ponds. A hybrid might have intermediate swimming ability, making it outcompeted in both fast and still water. Or think of a hybrid plant whose flowering time falls exactly between the peak flowering times of its parents, meaning neither set of pollinators is abundant when it blooms. The environment makes the hybrid unfit.
So its characteristics fall between two stools, ecologically speaking?
Exactly. But often, the problems are deeper, more fundamental. That's intrinsic isolation.
Intrinsic, meaning the problem is inherent to the hybrid itself, regardless of the environment.
Correct. Intrinsic barriers mean the hybrid has low fitness because of problems with its own development, physiology, or reproduction stemming from its mixed genetic makeup. These problems occur even in a benign or optimal environment. The hybrid might simply die early, reduce viability, or be unable to produce functional gametes, sterility.
And this intrinsic failure is caused by incompatible genes.
Yes, fundamentally. It's caused by incompatible interactions between the genes inherited from the two different parent species. The genetic instructions from the two lineages just don't work well together.
Okay, and this leads us to a really important theoretical model for how this incompatibility evolves, right? The Dobzhansky-Muller model.
The Dobzhansky-Muller incompatibilities, or DMIs. Yes, this is a cornerstone concept for understanding intrinsic postzygotic isolation.
Why is it so important?
Because it elegantly solves a major paradox. Reproductive isolation, especially intrinsic hybrid inviability or sterility, is obviously bad for the fitness of the individuals involved. So how could natural selection possibly favor genes that cause it?
Right. Selection should favor things that increase fitness. How does a fitness-reducing barrier evolve?
The DMI model shows how these incompatibilities can arise as an accidental byproduct of normal adaptive evolution occurring independently in two isolated lineages. Selection isn't directly favoring the incompatibility itself.
Okay, walk me through how that works. It involves interactions between genes at different loci, right?
Exactly. Imagine an ancestral population that has compatible alleles at two interacting gene loci. Let's call them A and B. The genotype might be AA$ or BAB80. Now, this population splits into two geographically isolated lineages, lineage 1 and lineage 2.
Okay, separated.
In lineage 1, a new mutation arises to the A locus, allele B$, or 1. Let's say $8 is beneficial in that environment. Perhaps it improves metabolic efficiency. Natural selection favors it and it eventually fixes, so lineage 1 becomes A1A1, B-Do, BBB. It works fine with the ancestral B$.
Okay, $8 good.
Meanwhile, completely independently over in lineage 2, maybe a different mutation arises at the B locus, allele B$. Let's say B$ is also beneficial in its local environment, maybe conferring disease resistance. It fixes, so lineage 2 becomes $8 B1B88. It works fine with the ancestral A deal.
So both lineages have improved their fitness independently by fixing different beneficial mutations at different genes.
Correct. $8 is fine with B$, and $8 is fine with B$. No problem so far. But now, imagine the geographic barrier disappears and individuals from lineage 1 and lineage 2 meet and hybridize.
Secondary contact. Okay, what happens in the hybrid?
The F1 hybrid inherits A dollars from the lineage 1 parent and A dollars from the lineage 2 parent, and D dollars from lineage 1 and B dollars from lineage 2. So its genotype is A1A dollar, B1 B dollar. The crucial point is that the allele A$1 is now interacting with allele E dollars for the first time ever within the same organism.
Ah, a novel combination that selection has never seen before.
Exactly. And the DMI model proposes that these novel combinations of alleles, A dollars with B dollars in this case, or potentially others involving homozygotes in later generations like A1, A1, B1, and B1, are often functionally incompatible. They might disrupt development, cause physiological problems, or lead to sterility. The two individually beneficial alleles clash when brought together.
So the reproductive barrier, the hybrid inviability or sterility, didn't evolve because selection wanted to create a barrier. It evolved because selection favored A dollars in one place and B dollars in another place, and their interaction just happened to be detrimental when they were eventually mixed. It's an unlucky accident of independent evolution.
That's the core idea. It explains how intrinsic postzygotic isolation can arise passively, without invoking selection for reduced hybrid fitness. And we have solid genetic evidence for these kinds of interactions.
Like the example in the plant Arabidopsis thaliana.
Right. The book mentions studies where researchers crossed different Arabidopsis strains that had compensated for the loss of function in one copy of an essential duplicated gene, a paralog, by relying on the other copy. For example, strain 1 might have a functional alpha gene, but a broken beta gene, alpha plus beta, while strain 2 has a broken alpha but a functional beta, alpha beta plus dial.
Both strains are fine on their own.
Yes. And the F1 hybrid, alpha beta plus beta dollar, is also fine because it has at least one working copy of each gene.
But then in the next generation, the F2.
When those genes segregate and recombine in the F2 generation, you can unfortunately produce offspring that inherit only the broken copies of both genes, alpha beta beta wall. That combination is lethal. It's a direct demonstration of a two-locus DMI causing hybrid inviability.
A very clear genetic clash. Okay, so DMIs involving nuclear genes are a major source of intrinsic isolation. Are there other types?
Yes. Another important category is cytonuclear incompatibility. This involves a mismatch between genes in the cell nucleus and genes in the mitochondria.
Mitochondria have their own DNA.
They do. A small circular genome inherited maternally in most animals. These mitochondrial genes encode components essential for energy production, but they have to work intimately with hundreds of proteins encoded by nuclear genes that are also involved in mitochondrial function or assembly.
So the nuclear genome and the mitochondrial genome have to co-evolve to remain compatible.
Precisely. As two populations diverge, their nuclear genes and their mitochondrial genes co-evolve along slightly different paths. If you then create a hybrid that combines the nucleus from one population with the mitochondria from the other,
things might not work properly, like putting the wrong software on the hardware.
That's a good analogy. The nuclear proteins might not interact correctly with the mitochondrial components, leading to reduced energy production, lower fitness, or even sterility. The copepod Tigriopus californicus is cited as a key example where hybrid fitness plummets when you mix mitochondria from one population with the nuclear background of another.
Another layer of potential genetic conflict in hybrids. And what about bigger structural changes, like to the chromosomes themselves?
Absolutely. Chromosome rearrangements are a major player in postzygotic isolation, particularly in causing hybrid sterility. Sister species often differ in the structure of their chromosomes, things like inversions, where a segment of a chromosome gets flipped, or translocations, where parts of different chromosomes get swapped.
How do these structural differences cause problems in hybrids?
The main issue arises during meiosis, the specialized cell division that produces sperm and eggs. For meiosis to work correctly, homologous chromosomes need to pair up precisely along their entire length.
But if one chromosome has an inverted segment compared to its homologue from the other parent,
they can't pair up properly in that region. They might form complex loops or other structures trying to align the matching parts. When these chromosomes then try to segregate into the daughter cells, the gametes, things often go wrong.
leading to unbalanced gametes.
Exactly. Gametes might end up with missing chunks of chromosomes or duplicated chunks. These aneuploid gametes are usually inviable or lead to inviable zygotes. The end result is that the hybrid individual, heterozygous for the rearrangement, produces few or no viable sperm or eggs. It's effectively sterile or has drastically reduced fertility.
Even if the hybrid itself looks perfectly healthy otherwise.
Right. The book mentions the common shrew, Sorex araneus, where different geographic races have accumulated different chromosome fusions. Where these races meet, there are very narrow hybrid zones.
Why narrow?
Because the hybrids formed in that zone, which are heterozygous for the different chromosome arrangements, have significantly lower fertility, sometimes reduced by 50% or more. Selection acts strongly against them, preventing the genetic differences from mixing broadly and keeping the races distinct. The chromosomal differences act as a powerful postzygotic barrier. Hashtag 2.4 speed of isolation.
That shrew example makes it sound like once you have strong postzygotic barriers like chromosomal incompatibility, they're really effective at maintaining separation. But how long does it typically take for these barriers, both pre- and postzygotic, to evolve? Is speciation fast or slow?
Well, the answer is, it varies enormously. The time required can range from practically instantaneous to millions and millions of years.
Instantaneous?
We'll touch on this more later, but speciation via polyploidy, especially in plants, can happen in literally 1 or 2 generations. A new polyploid individual is immediately reproductively isolated from its diploid parents. That's the extreme fast end.
Okay, so sometimes it's super rapid, but what about the more gradual accumulation of barriers, like the DMIs or behavioral changes we've been discussing?
That tends to be much slower, especially for the intrinsic postzygotic barriers. The accumulation of sufficient DMIs or chromosomal rearrangements to cause complete sterility often takes a very long time.
How long are we talking?
Studies comparing related species suggest that prezygotic isolation, things like differences in mating signals or habitat preference, often evolves considerably faster than intrinsic postzygotic isolation.
So behavioral or ecological walls go up faster than genetic incompatibility walls.
That seems to be a common pattern, yes. The book cites examples from birds, where even species that diverged maybe 5 million years ago can sometimes still be crossed in captivity and produce fully viable, fertile hybrids.
5 million years. That's incredible. They've been separate lineages for that long, adapting independently, yet their genomes haven't accumulated enough DMIs or structural changes to make hybrids inviable or sterile.
It's astonishing, isn't it? It really highlights that significant genetic incompatibility can be slow to evolve. It also suggests that in many animal groups, especially mobile ones, the barriers that are initially most important for divergence might be prezygotic ones, changes behavior, ecology, or mate recognition.
But once those strong postzygotic barriers do finally evolve, like complete hybrid sterility caused by massive genetic or chromosomal clashes, that seems like a point of no return. Right.
Very likely, yes. Once you have complete hybrid inviability or sterility, the genetic separation is essentially permanent and irreversible. That's when you can be pretty confident that speciation is complete and the 2 lineages will never merge back together.
Okay, we've established what reproductive barriers are and the different types, but we need to circle back to that fundamental question, the paradox. These barriers often reduce fitness, at least initially, for hybrids. So how do they actually evolve? Why would selection favor something that seems detrimental?
Right. The traditional answer, and certainly a very common scenario, relies heavily on allotry. The idea is that populations get geographically isolated first.
The physical barrier comes first.
Yes. Then, while they're separated, they diverge passively due to genetic drift and, more importantly, due to natural selection, adapting them to their different local environments. The reproductive barriers arise simply as an accidental byproduct of this independent divergence. They weren't selected for directly, they just happened as the populations changed in other ways.
So isolation happens by chance due to accumulated differences. That makes sense for allotry. But can speciation also be driven more actively? Can selection directly favor divergence that leads to isolation, even if populations aren't completely separated?
Absolutely. Biologists now recognize several major mechanisms where selection plays a much more direct role in driving the evolution of reproductive isolation. These are often categorized based on the type of selection involved. One of the most pervasive is ecological speciation. Hashtag 3.2 ecological speciation.
Ecological speciation. This sounds like the barriers arise as a side effect of adapting to different environments. Similar to the allopatric idea, but maybe happening even with some gene flow.
Exactly. The core idea is that divergent natural selection pressure, driven by differences in the environment or ecological niche, causes populations to adapt in different directions. And reproductive isolation evolves as a correlated response, a byproduct of that ecological adaptation.
The selection is primarily for survival or resource use in a specific environment, and the isolation kind of tags along for the ride.
Precisely. And we have compelling evidence for this, even from laboratory experiments. The book describes a classic study using Drosophila fruit flies.
What did they do?
Researchers started with a single large population of flies. They split it into several replicate lines. Half of the lines were then consistently raised on food medium based on starch, while the other half were raised on food medium based on maltose.
So just a difference in diet. That was the only selective pressure deliberately applied.
That was the key divergent pressure. They maintained these lines for many generations. I think it was only about 20 generations in some experiments, which is remarkably fast evolutionarily.
Okay, 20 generations adapting to either starch or maltose. Then what?
Then they tested whether any reproductive isolation had evolved between the starch-adapted flies and the maltose-adapted flies. They did mate choice experiments.
And did they find anything?
Yes. They found clear evidence of partial but statistically significant sexual isolation. Flies showed a tendency to prefer mating with other flies that had been raised on the same type of food.
Wow, just adapting to a different food source for 20 generations started to create a mating barrier.
It did. The index of isolation wasn't complete, but it suggested they had already moved a considerable way towards becoming reproductively isolated, purely as a result of adapting to different diets.
How does that happen? How does adapting to starch versus maltose affect who you want to mate with?
That's the crucial question. It implies there must be some genetic link between the genes involved in metabolizing the food source, which were under direct selection, and the genes influencing mate choice or mating cues, like pheromones, perhaps.
So maybe the same gene affects both digestion and mate preference. That's called pleiotropy, right?
Pleiotropy is one possibility. Or it could be that the genes for diet adaptation are located very close on the chromosome to genes involved in mating signals or preferences, so they tend to be inherited together. That's genetic linkage. Either way, selection acting on ecological adaptation dragged reproductive isolation along with it.
That's powerful. It suggests that just by adapting to different environmental challenges, populations can quite rapidly start down the path to becoming distinct species. It makes ecological speciation seem like it could be incredibly common.
It's thought to be extremely common, yes, especially when environments are patchy or populations colonize new niches. Adaptation drives divergence and isolation follows. Hashtag 3.3 speciation by genetic conflict.
Okay, so ecological selection is one major driver. What else can cause speciation? You mentioned internal conflict earlier, something about selfish genes. We touched on this way back in Deep Dive 5, I think.
That's right. Speciation doesn't always have to be driven by external environmental pressures. It can also arise from internal evolutionary arms races within the genome itself, driven by what we call selfish genetic elements or genetic conflict.
Selfish genes. These are genes that promote their own transmission, even if it harms the organism.
Exactly. They prioritize getting themselves into the next generation, sometimes at a cost to overall fitness. A prime example discussed in the context of speciation is the overdrive gene system found in some Drosophila species, like Drosophila pseudoobscura.
Overdrive, what does it do?
It's a fascinating, slightly sinister system. Overdrive is essentially a meiotic drive element located on the X chromosome. In males, who are XY, if they carry the overdrive X chromosome, it produces a factor that sabotages or kills the sperm carrying the Y chromosome during sperm development.
Seriously, it kills off the competition.
Effectively, yes. This means that nearly all the functional sperm produced by that male will carry the X chromosome with the overdrive gene, massively biasing inheritance in its favor.
That sounds incredibly selfish and potentially bad for the population if it skews the sex ratio too much.
It can be. And because it's detrimental, it creates intense selective pressure on the rest of the genome to evolve ways to counteract it. Populations typically evolve suppressor genes, often on the Y chromosome or autosomes, that shut down or resist the killing effect of overdrive.
So you get an arms race, the selfish gene trying to cheat, and the rest of the genome trying to stop it.
Exactly. Now imagine two populations become isolated. They might independently evolve slightly different versions of the overdrive system and different corresponding suppressor genes.
Okay, different weapons, different shields.
Right. Now what happens if these two populations hybridize? A hybrid male might inherit the overdrive gene from population A, but the suppressor genes from population B.
And the suppressor from B might not work against the weapon from A.
Precisely. The mismatch between the selfish element and its evolved suppressor can lead to catastrophic failure in the hybrid, often resulting in complete or near-complete male sterility.
Ah, So the genetic conflict itself, the arms race happening independently in isolation, directly creates a strong postzygotic barrier, hybrid sterility, when the populations mix.
Yes. It's a way to generate DMI-like incompatibilities potentially very quickly, driven entirely by this internal genetic warfare rather than external ecology. It's thought that genetic conflict might be a particularly rapid engine for the evolution of postzygotic isolation in some groups. Hashtag tag 3.4 speciation by sexual selection.
Okay, ecological pressures, internal genetic conflict. What about the choices animals make about their mates? Sexual selection seems like it could drive divergence really fast, too.
Absolutely. Speciation by sexual selection is considered another major pathway, especially in groups with complex mating behaviors. It works because sexual selection acts directly on the traits involved in reproduction itself, mating signals and preferences.
So if female preferences change in one population,
Then the male trait that satisfies that preference will be strongly selected for in that population. If a different preference evolves in another population, then a different male trait will be favored there.
and if those preferred traits are different enough,
The two populations might simply cease to recognize each other as potential mates. The divergent sexual selection directly creates a prezygotic barrier, sexual isolation.
because mate recognition fails.
Right. And this can happen even if the populations are ecologically very similar. The divergence is driven purely by arbitrary or runaway changes in mating preferences. The Hawaiian crickets, genus Lapala, are a fantastic example.
What's special about them?
There are many species on the Hawaiian Islands, often living in similar habitats, but they are strongly differentiated by the pulse rate of the male courtship song. And crucially, female preference is tightly tuned to the specific pulse rate of males from their own species or population.
So the song and the preference evolve together.
They co-vary strongly. If you take a female from one population and play her the song of a male from a genetically distant population with a different pulse rate, her response, her willingness to approach or mate, drops off dramatically.
So the divergence in the mating signal, driven by divergent female preferences, acts as the primary isolating mechanism. Sexual selection is building the wall.
It appears to be the main driver in that group. And perhaps the most famous example of speciation thought to be heavily driven by sexual selection is the incredible radiation of cichlid fishes in the African Great Lakes, like Lake Victoria and Lake Malawi.
Ah, yes, the cichlids. Hundreds of species evolving incredibly rapidly in the same lakes.
Exactly. Many of these species are ecologically quite similar, but they differ dramatically in male coloration patterns. And experiments show that females have very strong specific preferences for the color patterns of males of their own species.
So female choice based on male color is keeping the species distinct.
It seems to be a primary factor. This is often linked to what's called sensory drive, the idea that the environment influences what signals are most effective or easily perceived, which in turn shapes female preferences and male traits.
How does that work with the cichlids?
Well, the color of light changes with water depth and clarity. A color pattern that's vibrant and conspicuous in clear, shallow water might be dull or invisible in deeper, murkier water. So selection might favor different colors at different depths or in different parts of the lake based purely on visibility.
And female preferences track those locally optimal colors.
That's the idea. And it's supported by observations that when water clarity decreases, for example, due to pollution or sediment runoff, the visual distinctions between species break down.
What happens then?
Females can no longer reliably distinguish males of their own species from others based on color. The sexual isolation barrier weakens, and you often see increased hybridization and potential collapse of species boundaries. It really highlights how crucial sexual selection on visual cues is for maintaining species diversity in that system, and how sensitive it can be to environmental change. Hashtag track 3.5 reinforcement.
Okay, so we have ecology, genetic conflict, and sexual selection driving divergence and potentially creating barriers. But what happens if two populations have diverged in isolation, develop some level of incompatibility, maybe weak postzygotic barriers like slightly unfit hybrids, and then they come back into contact, secondary contact?
Right. This is where another process called reinforcement can come into play. Reinforcement is specifically about natural selection strengthening reproductive barriers, particularly prezygotic ones, after secondary contact has occurred.
Why would selection strengthen barriers after contact?
Because of the cost of hybridization. If the hybrids produced by mating between the two diverged populations have low fitness, if they're inviable, sterile, or ecologically disadvantaged due to those DMIs or other incompatibilities that evolved in allopatry, then individuals who mate with the wrong type are wasting their reproductive effort.
They're producing offspring that won't contribute much to the next generation. A genetic dead end, potentially.
Exactly. So under these conditions, there will be natural selection favoring any genetic variants that cause individuals to mate preferentially with their own type. Selection favors choosiness.
Ah, so selection actively favors stronger prezygotic isolation to avoid producing those low-fitness hybrids in the first place.
Precisely. Reinforcement acts to build up barriers like differences in mating signals, stronger mate preferences, or shifts in flowering time, anything that reduces the frequency of those costly interpopulation matings. It generally doesn't act directly on postzygotic barriers because the fitness cost there is only realized after the mating has already happened. Reinforcement is about preventing the mistake beforehand.
Makes sense. Do we have examples where this seems to have happened?
The textbook example involves phlox flowers in Texas. There are two species, Phlox tremondi and Phlox cuspidata.
Okay.
Where these two species grow separately in allopatry, they both typically have light blue flowers. Easy to confuse.
Both blue when they're apart.
Yes. But in areas where their geographic ranges overlap in symmetry, something interesting happens. P. drumondii populations have evolved striking dark red flowers instead of blue.
A dramatic color shift only where they meet the other species. Why?
It's strongly suspected to be reinforcement. Crosses between blue P. drumondii and P. cuspidata produce hybrids that have significantly reduced fertility up to 90% lower pollen viability in some cases. That's a huge fitness cost to hybridization.
So selection penalizes individuals that hybridize.
Heavily. The hypothesis is that the shift to red flowers in sympatric P. drumondii was favored because it makes them visually distinct from the blue P. cuspidata. Pollinators, especially butterflies, which are key pollinators for phlox, are less likely to move pollen between the very different red and blue flowers compared to moving pollen between two similar blue flowers.
The color difference enhances pollinator fidelity, reducing the chance of costly hybridization.
Exactly. The strong postzygotic incompatibility, low hybrid fertility, provided the selective pressure and the evolution of the prezygotic barrier, different flower color controlled by just a couple of genes, was the response. It's a beautiful example of selection reinforcing reproductive isolation to prevent wasted reproductive effort.
Okay, we've talked about what barriers are and why they might evolve: ecology, conflict, sexual selection, reinforcement. Now we need to put this into a spatial context. How does the geographic arrangement of populations influence the speciation process?
Right, the geography of it all. The book lays out three main modes based on the level of geographic separation and gene flow during divergence. That's right. These modes basically describe speciation relative to the potential for genes to flow between the diverging populations. They're illustrated nicely in figure 9.22.
Let's outline them. What's the first and probably most intuitive one?
The first is allopatric speciation. Allo meaning different, patric meaning homeland. This is speciation that happens when populations are in complete geographic separation. There's effectively zero gene flow between them because of a physical barrier.
Like the mice on opposite sides of the canyon we discussed earlier, or populations on different islands.
Exactly. This is widely considered the most common mode of speciation, especially in animals. The physical isolation comes first, then divergence happens independently.
Okay, allopatric means total separation. What's the next mode?
Next is parapatric speciation. Para meaning beside or near. Here, speciation occurs between populations that are geographically adjacent. They have ranges that abut, but they don't overlap extensively.
So they're neighbors, but distinct populations. Is there gene flow?
Yes. Crucially, there is some potential for gene flow between them, typically localized right along the boundary where the populations meet. This often forms what's called a hybrid zone.
Okay, so adjacent ranges with some gene flow across the border.
Right. And the third mode is sympatric speciation. Sym meaning same. This is speciation occurring within a single geographic area. The diverging populations overlap completely, and all individuals are, initially, potentially capable of encountering and mating with each other. Gene flow potential is initially very high.
So no geographic separation at all. Divergence happening right inside the ancestral population's range. That sounds difficult.
It is generally considered the most difficult and probably the least common mode, precisely because of that high potential for gene flow, which constantly works to counteract divergence.
Okay, so allopatric, separate, parapatric, adjacent with some gene flow, and sympatric, overlapping with high gene flow. Let's dig into each of these a bit more. Hashtag 4.2, allopatric speciation. Allopatric speciation seems like the standard model. A population gets split by some physical barrier.
Could be glaciation advancing and retreating, a mountain range uplifting, a river changing course. sea level rise, isolating islands, continental drift. Lots of geological or climatic events can fragment populations.
And once they're separated, no more gene flow. They're on their own.
Correct. Then divergence happens. This can be driven by genetic drift, especially if one or both of the isolated populations are small. Random changes in allele frequencies can accumulate differently in the two groups.
Drift just makes them different by chance over time.
Right. But perhaps more importantly, if the environments in the two isolated areas are different, natural selection will drive local adaptation. Each population adapts to its own specific conditions, different predators, different food sources, different climate.
And these adaptations accumulating over time
Can lead, often accidentally as we discussed with DMIs, to the evolution of reproductive isolating barriers. Maybe adaptations for diet, change mating pheromones as a byproduct, or adaptations to climate alter breeding season timing.
So, if and when that physical barrier eventually disappears and the two populations come back into secondary contact,
They might have diverged so much that they no longer recognize each other as mates, or they produce unfit hybrids. Voila, allopatric speciation is complete.
and we have evidence linking the amount of divergence to the time spent apart.
Yes, quite a bit. The book references studies on the lungless salamander, Desmognathus acrophaeus, in the Appalachians. Different populations are isolated in different stream drainages.
Okay.
When researchers conduct mate choice trials in the lab, they find that the strength of sexual isolation, how much females prefer males from their own population versus males from other populations, is strongly correlated with the geographic distance separating those populations in nature.
So greater distance implies longer isolation time.
Generally, yes. Distance serves as a proxy for time since divergence. The longer they've been apart, the more genetically different they become, and the stronger the reproductive barriers, in this case sexual isolation, tend to be. It supports the idea that divergence accumulates gradually during allopatric isolation.
And this model also reinforces the idea that preventing gene flow is critical. Speciation should be harder if gene flow is high, right?
Absolutely. Allopatric speciation works because the physical barrier stops gene flow. If gene flow is high, it constantly mixes alleles between populations, preventing them from diverging significantly.
So it expects speciation to be easier in organisms that don't move around much.
Generally, yes. The book shows data, figure 9.24, suggesting that speciation rates are often higher in taxa with limited dispersal ability, like land snails, compared to highly mobile taxa, like birds or bats, which can maintain gene flow over larger distances. Similarly, speciation is often more likely on smaller, more isolated islands where gene flow from the mainland is cut off. High gene flow is the enemy of speciation. Hashtag tag 4.3 parapatric speciation.
Okay, allopatry makes intuitive sense. But what about parapatry? Divergence between adjacent populations with some gene flow leaking across the boundary? How can they become distinct species if they're constantly swapping genes?
That's the key challenge for parapatric speciation. For it to work, the divergent selective pressures across the environmental boundary must be incredibly strong enough to counteract the homogenizing effect of the gene flow that's occurring.
So you need a really steep change in the environment, a sharp selection gradient.
Exactly. Imagine a transition from, say, normal soil to soil contaminated with heavy metals over a short distance. Plants living on the contaminated soil face intense selection for tolerance, while plants just meters away on normal soil face selection against tolerance because it might be costly.
And selection is pulling the populations in opposite directions right across that border.
Right. And if selection is strong enough, it can maintain genetic differences between the adjacent populations, despite the gene flow trying to mix them up. This often results in a narrow hybrid zone where hybrids might be formed, but they're often less fit than either parent type because they're not perfectly adapted to either side of the gradient.
Selection against hybrids in the middle helps keep the populations distinct.
It can contribute, yes. A fantastic natural example of parapatric divergence driven by strong ecological selection is the lizards living in and around the white sands gypsum dunes in New Mexico.
Ah, the white sands again. We mentioned them earlier with adaptation.
Yes, because it's such a striking case. These dunes are geologically very young, formed less than 5,000 years ago. They are brilliant white gypsum sand surrounded by much darker reddish-brown desert soils.
A stark environmental contrast.
Extremely stark. Several lizard species live both on the dunes and in the surrounding dark soil habitats, including the fence lizard, Celoporus undulatus, and the little striped whiptail, Aspidoscelis inornatus. Actually, the book mentions the earliest lizard, Holbrookia maculata, as another example.
Okay, and the ones on the dunes are white.
They've evolved incredibly pale, almost white coloration. It's camouflage crypsis driven by intense predation pressure from birds. Lizards that don't match the background get picked off very quickly.
So survival selection is extreme, right at the boundary between white sand and dark soil.
Absolutely. Selection is favoring white lizards on the sand and dark lizards just off the sand. And what's fascinating is that this strong selection for color seems to be driving reproductive isolation as well.
How so?
Well, studies using genetic markers show surprisingly sharp genetic differentiation between the dune populations and the adjacent dark soil populations right across that habitat boundary, despite the fact that lizards could easily move back and forth.
So gene flow isn't homogenizing them completely.
No. Selection is apparently strong enough to maintain the genetic differences. And in some cases, the genes involved in coloration might also affect other traits, perhaps related to mating signals or preferences. For example, changes in body color might affect the appearance of ventral patches used in courtship displays.
So adapting to the local background color accidentally creates a barrier to mating with lizards from the other background.
That's the idea behind parapatric ecological speciation.
Yeah.
The strong divergent selection across the environmental gradient acts like a barrier, preventing widespread gene flow and allowing the adjacent populations to differentiate, potentially leading to full species status, even while they remain in contact. Hashtag tag 4.4 sympatric speciation.
Which brings us to the most controversial mode, sympatric speciation. Divergence happening within the same geographic area with potentially high gene flow. This seems like swimming upstream against a strong current of genetic mixing. How can it possibly work?
It is definitely the most challenging scenario theoretically. The big problem, as you said, is recombination. You have genes causing adaptation to, say, resource A, and other genes causing individuals to prefer mating with others who use resource A.
Yeah.
In a freely mixing population, recombination will constantly break apart that association. Offspring might inherit the genes for using resource A, but the genes for preferring mates who use resource B, or vice versa. This prevents the buildup of coordinated adaptation and assortative mating needed for divergence.
So gene flow and recombination just scramble any incipient differences.
They make it very difficult. For sympatric speciation to occur, you generally need very specific conditions. You need really strong divergent selection, favoring different ecological niches or resources within the same area.
Strong selection, pulling the population in two different directions simultaneously.
Yes. And crucially, you often need traits that directly link that ecological divergence to reproductive isolation. These are sometimes called speciation traits or magic traits.
Magic traits, what does that mean?
It refers to traits where the same gene, or tightly linked genes, affect both ecological adaptation and mate choice, or reproductive timing. This creates an automatic association between adapting to a certain niche and mating with others adapted to that same niche, helping to resist the homogenizing effects of recombination.
Okay, so the adaptation and the isolation are inherently coupled. Can you give an example?
The absolute classic textbook example of sympatric speciation driven by this kind of linkage is the apple maggot fly, Raguletus palmanella.
Right, we mentioned this fly earlier too. Tell us the story again in the context of symmetry.
Okay, so historically, Raguletus palmanella in North America exclusively laid its eggs on the fruits of native hawthorn trees. The larvae developed inside the hawthorn fruit.
Hawthorn was its ancestral host.
Yes. But then, about 150 years ago or so, domestic apples were introduced and became widespread in North America. Apples are related to hawthorns, but are a distinct host. At some point, a portion of the Raguletus population started successfully laying eggs on apples.
A host shift. Okay.
Yeah.
How does that lead to sympatric speciation? They're still flying around in the same orchards, right?
They're in the same orchards, geographically sympatric. But here's the key. Raguletus flies tend to mate on or near the fruit where they themselves emerged.
Ah, so if you grow up on an apple, you tend to find a mate on an apple tree.
Exactly. And if you grow up on a hawthorn, you tend to mate on a hawthorn tree. This creates immediate ecological isolation based on host preference. The apple flies primarily encounter and mate with other apple flies, and hawthorn flies with other hawthorn flies.
So the host choice itself acts as a mating barrier.
It acts as a strong component of one, but there's another crucial factor: timing. Apples ripen several weeks earlier in the summer than hawthorn fruits.
Different fruiting times.
Right. This difference in timing created strong selection pressure on the flies, using apples to shift their life cycle earlier. Apple flies needed to emerge from their overwintering pupal stage earlier in the season to match the availability of ripe apples. Hawthorn flies continued to emerge later, timed for hawthorns.
So the host shift also drove divergence in their emergence time, their seasonal timing.
Yes, and that difference in emergence time creates a powerful temporal isolating barrier, further reducing gene flow between the apple-associated flies and the hawthorn-associated flies, even though they live in the same geographic locations.
Wow. So the single ecological shift using apples instead of hawthorns simultaneously caused both ecological isolation, mating on the host, and temporal isolation, different emergence times. That sounds like the magic trait coupling you mentioned.
It's a perfect example. The choice of host ecology is directly linked to when and where you meet, reproduction. This strong coupling allowed the apple and hawthorn host races to diverge genetically and reproductively in symmetry, despite ongoing gene flow, which is measurable but reduced. They are now considered by many to be well on their way to becoming fully distinct species, all within the last century and a half. It demonstrates that sympatric speciation, while challenging, is definitely possible under the right conditions.
We've covered the main geographic modes, but the book also highlights a couple of special, often very rapid mechanisms of speciation. The first one you already mentioned briefly, polyploidy. Hashtag tag 5.1 rapid speciation by polyploidy.
Right, polyploidy. This deserves special mention because it's a major route to speciation, particularly in plants, and it's fundamentally different from the gradual accumulation of barriers we've mostly discussed.
Remind us what polyploidy is again?
It's the condition of having more than two complete sets of chromosomes. Most animals and many plants are diploid, 2N, meaning they have two copies of each chromosome. Polyploids might be tetraploid, 4N, hexaploid, 6N, or even higher. It's essentially a duplication of the entire genome.
A massive genetic event, and you said this is common in plants.
Extremely common. Estimates suggest that perhaps 15% of speciation events in flowering plants, and maybe even more in ferns, involve polyploidy.
And how does it cause speciation so rapidly? You said potentially in one generation.
It creates almost instantaneous reproductive isolation. Let's say a diploid plant, 2N, produces some unreduced gametes, also 2N instead of the usual N, due to an error in meiosis. If two of these 2N gametes fuse, or if a 2N gamete fuses with a normal N gamete and the resulting 3N offspring somehow undergoes genome doubling, you can get a tetraploid 4N offspring.
Okay, a 4N individual appears in a 2N population.
Now, what happens if that 4N individual tries to mate back with the original 2N parental population? The offspring will be triploid, 3N getting 2N from the tetraploid parent and N from the diploid parent.
Triploid. Is that a problem?
Usually, yes. Triploids often have major problems during meiosis because they have three copies of each chromosome that can't segregate evenly into gametes. This typically results in very high sterility. Think of mules, horse donkey hybrids, or seedless watermelons, which are often triploid.
So the new tetraploid can't effectively interbreed with its diploid ancestors. It's reproductively isolated right away.
Exactly. It can generally only reproduce successfully by mating with other tetraploids or by self-fertilizing, if the plant is capable of that. It forms its own reproductively isolated lineage almost instantly.
And this happens sympatrically, right within the parent population.
Yes, it's a classic mode of sympatric speciation. It doesn't require geographic separation. It can happen spontaneously. We distinguish between autopolyploidy, where the duplicated chromosome sets all come from the same species, and allopolyploidy, which is even more common and involves hybridization between two different species, followed by genome duplication.
Hybridization first, then doubling.
Right. This combines the genomes of two species and then makes the hybrid fertile by giving every chromosome a proper pairing partner for meiosis. Many important crops, like wheat, cotton, and coffee, are ancient allopolyploids, and the book mentions recent examples like the goat's beards, Tragopogon, in North America.
Goat's beards?
Yes, 3 diploid Tragopogon species were introduced to North America from Europe. Within the last century, they have hybridized, and 2 new, distinct allotetraploid species have formed spontaneously and become established in the wild. We've literally watched new species arise via polyploidy in historical times. Hashtag 5.2 peripatric speciation, founder effect.
Okay, polyploidy is instant speciation. What's the other special case mentioned? Peripatric speciation. It sounds a bit like allopatric.
Is closely related to allopatric speciation, yes. Peripatric speciation, peri meaning around or peripheral, is basically a specific scenario of allopatric speciation that involves the founder effect.
Ah, the founder effect. When a new population is started by a very small number of individuals, we covered that under genetic drift.
Exactly. So, peripatric speciation occurs when a small group of individuals buds off from the main population and colonizes a new isolated location, perhaps on the periphery of the species range, like a few birds getting blown off course to a remote island.
A founder event establishing a new isolated population.
Right. Because the founding group is very small, it likely carries only a fraction of the genetic diversity of the original large population, and possibly some rare alleles might be present at unusually high frequency just by chance.
So the starting gene pool is small and potentially non-representative.
Yes. And in this small, isolated founder population, genetic drift can be incredibly powerful. Random fluctuations in allele frequencies can cause rapid genetic divergence from the parent population. Additionally, the new environment might exert strong, novel selective pressures.
So intense drift plus potentially strong selection acting on a limited gene pool.
Can lead to very rapid divergence and the evolution of reproductive isolation. Ernst Mayr, who also championed the BSC, proposed parapatric speciation as a major mechanism for explaining the origin of highly localized distinct species, particularly on islands. He used the example of the paradise kingfishers, Tanysiptera, on islands near New Guinea.
Kingfishers.
Yeah, many small islands around New Guinea have their own unique, morphologically distinct form of Tanysiptera, often differing dramatically from the mainland species and from forms on other nearby islands. Mayr argued that these likely arose rapidly via founder events. A few individuals colonize an island, intense drift and selection drive rapid change, leading to a new species in isolation. It's essentially allopatric speciation accelerated by the potent effects of founding a population with very few individuals. Hashtag 5.3, the genomics of speciation.
Okay, so we have these different modes and mechanisms. To wrap things up, let's touch on how modern technology, specifically genomics, is changing how we study speciation. We're moving beyond just observing patterns to looking at the actual genes involved.
Absolutely. The field of speciation genomics is incredibly exciting. The goal now is to use whole genome sequencing data to pinpoint the specific genes, the speciation genes, that are actually responsible for causing reproductive isolation between diverging populations or species.
Finding the genes that build the walls.
Exactly. We want to know how many genes are typically involved. Are they clustered together or scattered across the genome? What are their functions? Do certain types of genes contribute more often than others? Genomics gives us the power to start answering these questions directly.
How do you find these speciation genes in a sea of potentially millions of base pairs? Especially if speciation is happening with some gene flow, like in parapetry or sympatry.
That's where one of the key concepts of speciation genomics comes in, looking for genomic islands of speciation.
Genomic islands, like islands of difference in an ocean of similarity.
Precisely. The idea is that if two populations are diverging but still exchanging some genes, speciation with gene flow, recombination in gene flow should tend to homogenize most of the genome over time. Alleles get mixed back and forth.
So most of the genome should look fairly similar between the two populations.
Right, except in the specific regions of the genome that contain the genes under strong divergence selection, the genes causing ecological adaptation to different niches, or the genes involved in mate choice, or the genes causing DMIs.
Selection is acting strongly to keep those specific regions different, even while the rest of the genome gets mixed up.
Exactly. Those regions containing the speciation genes resist the homogenizing effect of gene flow because selection is constantly favoring different alleles in the two populations. So if you compare the genomes of the two diverging populations,
you should see generally low levels of genetic differentiation across most chromosomes.
But then you'll find distinct islands or peaks of exceptionally high differentiation right around the locations of those key speciation genes.
You measure this differentiation using statistics like FST$.
Yes. FST is a common measure of how genetically different two populations are at a specific locus or across a genomic region. In the scenario of speciation with gene flow, we expect to see a pattern of low FST across most of the genome, punctuated by sharp spikes of high FST at these genomic islands.
That's the signal. The islands are where the action is, where the genes driving divergence and reproductive isolation reside.
That's the interpretation. Finding these islands allows researchers to zoom in and identify the candidate genes within them, and then test their functions to see if they really do contribute to ecological adaptation or reproductive barriers. It's a powerful way to dissect the genetic basis of speciation, moving beyond geographic models to the actual molecular changes involved. Hashtag tag outro.
Wow. Okay, this has been a truly fundamental deep dive, hasn't it? We've really pulled together all the genetic mechanisms, selection, drift, gene flow that we've been building up throughout Unit 2 and seen how they culminate in the origin of new species.
It really is the culmination. We started by challenging the simple idea of defining species by appearance and landed on the biological species concept, the idea that species are defined by reproductive isolation. by the inability to exchange genes freely.
And we saw that this isolation isn't usually one thing, but layers of biological walls, the reproductive isolating barriers.
Right, from the often faster evolving prezygotic barriers like differences in timing, habitat, mating signals, or gamete recognition, which prevent costly hybridization upfront.
To the deeper, often slower evolving intrinsic postzygotic barriers, like the fascinating Dobzhansky-Muller incompatibilities, where individually beneficial genes clash fatally in hybrids, or chromosomal rearrangements that cause sterility. These build the permanent walls.
And we explored why these barriers evolve, often as an accidental byproduct of adaptation and isolation, allopatry, but also driven actively by ecological selection, by internal genetic conflict, or by divergent sexual selection, and even strengthened by reinforcement when unfit hybrids occur.
And finally, we placed it all in geographic context, allopatry being the most straightforward, but parapatry being possible with strong selection across boundaries, and even sympatry occurring under special circumstances, like with host shifts in apple maggot flies, or instant speciation via polyploidy in plants.
I think the biggest takeaway, really, is that speciation isn't typically a single event, but a process. It's often gradual, sometimes messy. It represents this dynamic tension between the forces driving divergent selection and drift, and the force trying to pull things back together, gene flow. Species boundaries can be fluid, especially early on, but they solidify as those reproductive barriers become stronger and more numerous.
A fantastic summary. And with that, we have officially concluded Unit 2 of our exploration of Futuyma and Kirkpatrick's evolution, focusing on how evolution works.
We've covered the core mechanism.
We have. So next time we embark on Unit 3, where we shift focus to the incredible results of these mechanisms, the products of evolution, what natural selection has wrought.
Looking at the amazing diversity and complexity that evolution has produced.
Exactly. And we're kicking off Unit 3 by tackling one of the most profound and in some ways puzzling products of evolution imaginable, the evolution of sex itself.
Ah, yes, sex. Why does it even exist? From a purely reproductive efficiency standpoint, cloning seems much simpler and faster. Why go through all the trouble of finding mates, meiosis, recombination?
The famous twofold cost of sex. It throws up huge evolutionary questions about its advantages and disadvantages. Why is it so widespread despite its costs?
It's one of the biggest questions in evolutionary biology, and it has generated decades of debate and research. We'll dive into the leading hypotheses next time.
I cannot wait to unpack that one. It should be fascinating. Join us then. Thanks for listening to Bookfulness.